Okay. Hi, I'm Jason Underwood, and I'm a principal scientist for PacBio, where I focus mainly on RNA work. And these days, most people are talking about a 30 kilobase long RNA called SARS-CoV-2. But today I'm going to tell you about our efforts to sequence full-length transcripts, but with single-cell information encoded into the sequencing reads. And so our real motivation for this is watching what's been going on in the biological community and the applications of single-cell techniques to many different aspects of biology and even medicine. And so the theme that we're seeing that goes through many of these studies is that no matter what technology here, whatever single cellulizer you're using, the main theme is to increase the number of reads and increase the number of cells that are assayed at one time. And so this is a clustering that shows 2 million mouse cells that have been evaluated at one time. But the theme that does seem to run through these studies as well is that there's a measurement of gene expression. And one thing we know is that it's not just genes that are being expressed in mammalian cells, but isoforms. So most genes make multiple isoforms. And so in that case, we want to be able to study that information with that kind of full-length context. And so this is an example showing 3 different isoforms made from one gene. These might have different cellular localization or a dominant and negative effect. They could have completely different functions in the cell. But when they're measured by common methods, in this case 3' tagging, such as the 10x or scRNA-seq methods, just a short tag is tagged at the 3' end of the transcript. And this is similar for what I call transcriptional unit tagging, where a transcript is evaluated by short reads that span along different spots on the transcript and even sometimes in introns. And then occasionally you catch reads that do span an exon-exon junction, but you don't necessarily have the information to stitch together a full-length RNA that can be assayed or actually assigned to what protein it would generate in the cell and thus what function it would serve. So we decided to approach this problem using PacBio's approach to RNA sequencing, which we call ISO-SEQ or isoform sequencing. So ISO-SEQ takes advantage of the full-length RNA information by generating a full-length cDNA that goes into our sequencing technology, which can generate long, accurate reads. And so there's no assembly required during the evaluation. Rather, you just get a full-length sequence. So the engine behind ISO-SEQ is sort of performing reverse transcription from RNA. So poly-RNA is reverse transcribed from its 3' end, and a template switch oligo is used to generate a sequence of a known sequence at the 5' end. And now this is capable of going into PCR, and we can generate what we call a SMART-Bell library. So this is a covalently closed piece of DNA that can go into our sequencing instrument. And so the way our sequencing works is we generate very long polymerase reads where the polymerase goes around and around this SMART-Bell and generates reads on both strands. And then once these are aligned to each other, we can generate what we call a hi-fi read. So our runs with ISO-SEQ can generate around 4 to 5 million full-length reads, and these are over 99% accurate. So these are very nicely capable of doing many things in the bulk level, and we've seen many examples of using that for studying biology and for doing annotation of genomes. But with this kind of accuracy, we thought we might be able to now apply this to single cell as well. So a normal ISO-SEQ library, as depicted in the upper left, has a 5' primer, the transcript sequence, a poly-A tail, and a 3' primer. On the right, you see a single-cell cDNA, and so this is many platforms that use this actually take advantage of a very similar mechanism to generate cDNA with the template switch oligo and PCR. However, many platforms encode additional information at the ends of the transcript, like a unique molecular index UMI, or a cellular barcode. And so we thought, well, can we take the same information and from these reads from a single cell platform and go ahead and call what we would call this UMI and barcode information. And so this plot shows you that on the x-axis is where we rank all the barcodes in a single cell sequencing run that came from the 10x platform with the human cell line K562 in conjunction with Mount Sinai. And then on the y-axis is showing the Illumina barcode rankings. And so we identified 2,555 cells that were sort of thought of as the cells of interest, so had enough reads in the 10x data on Illumina. And we found all of these exact same barcodes in the PacBio data as well. And this is by directly reading the FASTA information out of the sequencing. It's not by looking at the barcodes off of each other. So we thought this is something that we could go ahead and use to go ahead and use PacBio to study single cell libraries that were already being generated by people that were doing these kinds of things on a short read platform and getting the 3' information, for example. So I'm going to talk about a couple different platforms that we've used for this kind of analysis. And one that I'm not going to talk about that I've used personally, but has been used a couple times in the field, is using the 10x Chromium 3' solution, combining this with long reads. So one of these papers is from Gupta et al. From Hagen Tilgner's lab at Cornell, where they used the Chromium instrument to study mouse brain development. And then the other is from a group at the Earlham Institute, led by Ian McCauley. And Laura Mincarelli's paper showed analysis with both iso-seq and short reads, studying mouse hematopoiesis. And so just a couple snippets from that data that was quite exciting, I think, from the Hagen Tilgner's lab this shows this graphic from the paper from the gene BIN1. And so you can see that these individual tracks are showing the reads that are generated from individual cell types. So these are neurons in the top rows, and then astrocytes, glia, and oligodendrocyte precursors on the lower half. And if you look at those orange exons on the right, you can see that they're often included in the neural transcripts. But when you go down into the astrocytes, glia, and oligodendrocyte precursors, you see that these are not included as often. And if you look at the gen code annotations below, these are known to be alternative splicing events. But now with this technique, they could take advantage of this information to track which isoform is in which cell. And the brain's an extremely exciting place to do this because there's such diverse alternative splicing and cool biology behind that. And then second, I think this paper that just came out last week from the Earlham Institute has the combined single cell from short and long reads. And they were studying young mice and aging mice and looking at what their stem and progenitor cells were doing in the bone marrow. So what's happening in hematopoiesis in these? And a really interesting result that I thought what came out of this is that they saw that in the IGKC locus, so this is an immunoglobulin gene, that they could see that in aging mice, the amount of IGKC expression in short reads was quite increased. So that's the red here versus the blue. But when they went over and looked at the same data with the long reads, they could actually see something far more interesting, I think. So at the top of this graph, you see the constant regions are at the right side of the IGKC gene. And of course, these undergo VDJ recombination in the organism. So this is a rearrangement of the DNA. So you can see in the young mice, there was a pretty simple set of these sort of germline IGKC transcripts. But when they look in the aging mice, they saw a far larger diversity. So it wasn't just that they were studying that the gene went up. they actually were studying that the gene actually diversified quite significantly. And remember that with the 3' end only information, they would not have uncovered this kind of phenomena. So we've also taken on a few different platforms at PacBio through collaboration. And so I'm going to speak to you about a couple of those and cover some of that data. So one of those is the DROP-SEQ technique. So this is another microfluidic technique similar to 10x, but instead of doing reverse transcription in emulsion, the emulsion is broken and reverse transcription is done just in a test tube. And so we tried this using Dolomite's product, the NADIA. And so this may look like a panini press, but it's actually a single cell instrument that does DROP-SEQ. And so at first we had the Dolomite folks make us some libraries and we took a look at these. And so when we did that, we could see sorry, let me back up and say that we wanted to see, okay, we know these things make full-length transcripts, probably because of the template switch oligo technique, but are these actually looking like full-length transcripts? So in the effort to sort of walk before we run, we wanted to do a relatively easy experiment and then look at what those reads look like. And so we then took the long reads and classified them against reference annotations. And so I'll just cover that quickly using the SCONTI tool that was developed by Anna Canessa's lab. This lets you take long read iso-seq data and compare it against a reference annotation. And so a reference can then be compared to all of the long reads, and you might find full splice match, meaning we see the reference perfectly, the full length, including all the splice junctions. Incomplete splice match, meaning it matches partially. Or novel in catalog means that we've seen these exons before, but we've never seen this particular splicing event. Or novel not in catalog means we haven't seen one of these exons, so we have a new junction that we've never seen before. So we had the Dolomite folks ran what is colloquially called a barnyard mixing experiment, and that's to match mouse and human cells together in the microfluidics, and then carry out PacBio, and they performed Illumina sequencing as well. And so you can see on the top here with 2 different samples, we saw that many of the human transcripts were a full splice match, and we also saw that of the mouse ones. So it's really nice that mouse and human have pretty deep annotations. And then we saw incomplete splice match and these novels that showed up at the lower levels. And this is very similar to what we see when we take a high-quality RNA sample and perform bulk iso-seq. So we were quite encouraged by these results. But of course, we didn't want to do something with those cell lines. We wanted to do something a little more interesting. So actually took advantage of a long-time collaboration I've had going on with Evan Eichler's group and Alex Pollan at UCSF. And that's to study the great apes and what differences they harbor to make that make humans different than the great apes. And so because it's difficult to work with real ape samples, they've been using cerebral organoid models. So this is where you take iPS cells and push them towards neural fates in a dish, and then we could dissociate these and go ahead and assay these to do single cell iso-seq and short reads with them. And this was a follow-on to sort of the ape genome work by Cronenberg et al., and then Alex Pollan's work to establish cerebral organoids as a model for ape brain development. So we carried out the iso-seq on dissociated cerebral organoids that are about a month old. And we then sequenced these libraries on the PacBio Sequel 2. At the time, this was the V1 chemistry, and so the V2 chemistry would increase all of these stats by quite a bit. And once we did that, we took those reads and ran them through a few filters to sort of clean up some of the things that we know can happen in these types of libraries. And SCONTI can do these types of things. So one is to look for genomic poly-A priming. So we know, if anyone's familiar with single cell, they know that sometimes short stretches of A's can make the RT take off at that point rather than the legitimate poly-A tail. So we take out those types of events, and then there's some known RT switching effects, and then also we take out things that have unannotated non-canonical junctions. And so as shown on the bottom, we sort of know what the signals look like at the bottom or at the ends of the introns in messenger RNAs. So we look for the GU or AG or the AU and AC splice sites to filter those things out. And so then what we got out of this is we could look at not just the number of genes you see in these samples, and so that was around 14,000, but we could actually look now at unique isoforms. And so we got about 4 times the number of isoforms from these cerebral organoids than you would have gotten from just counting the genes. So remember that the 3' end type of product that the Dolomite NADIA usually runs using the DROP-SEQ workflow runs a shearing step that would just record that 3' information. But now with the full-length information, we can actually record isoform level information. So one of the aspects that we wanted to take a look at is the number of genes detected per cell. And so that as quite a bit more genes are detected per cell in short read data, but with long read data, we can still get a pretty good number by not increasing the cells to too many. So if you look at just the top 100 cell barcodes, we see about 729 genes detected per cell. If we look at the top 400, we see about 315. Now remember that there's about 4 times as many of these isoforms. So it's sort of a different counting than we would do for normal single cell biology. So one of the things we wanted to get out of these cerebral organoids is this also generating something that looks like a real transcript? So we went through some of the same steps we did with the experiments I described with the barnyard, but taking a look at reference annotations, because GenCode version 29 is quite good for human annotations. And so again, we compared what a reference annotation looked like to the transcripts we got out of the cerebral organoids, and we could see a really nice size distribution of the full splice match and incomplete splice match transcripts being the highest level, and these stretch all the way out to the 5 or 6 kb mark. And so some of these are not capturing some of the very longest genes we often capture with the bulk iso-seq method, but we still felt really encouraged by the level of profiling that we could do. But are these transcripts real? And so we wanted to dig into this more. And so for one, we wanted to look at what is the poly-A signal? What does the poly-A look like? And so one of the things we could do is look for the motif in the mRNA that looks like the poly-A signal. And so there's a poly-A signal upstream of the actual poly-A tail that directs cleavage and polyadenylation. And so we looked at what's the most common 6 nucleotides that are in that region just upstream of the poly-A tail. And we saw about 60% of them were the AAU-AAA, and second was AUU-AAA, about 16%. And remarkably, we look back at the literature, which is pre-next generation sequencing from 2000, and the top hexamers were these exact ones at 58% and 15%. So we felt quite encouraged that we're calling what looks to be real polyadenylation. Then we traveled to the other end of the transcript, and we said, can we take advantage of known 5' end data? And so this is a type of data called CAGE. And so we looked at the human full splice match, and we could often see that there's a CAGE peak that's within 50 base pairs of where we see our transcription start site in the long reads. So this shows that reverse transcriptase is getting all the way to the other end of the transcript during these single cell protocols with the DROP-SEQ, and that was very exciting. I thought it was really cool that even in the novel nodding catalog, so these are transcripts that we haven't seen before with new exons or new junctions, even those tend to have a CAGE peak that are within 50 base pairs, 67% of those. So that's exciting because this could be new biology that's in these cerebral organoids that now at single cell level you can start to dissect. But what about the middle? So what we did is take short reads to do splice junction validation. And so 87% of our splice junctions could be validated using GenCode version 29. And that makes some sense since many of these RNAs are abundant. But 13% being novel seemed kind of high. So what we did is said, you know what, version 29 probably hasn't done cerebral organoids. So we took advantage of some short read data sets from the human developing cortex, and we could find 3.8% more of those junctions. And then we took advantage of a gigantic data set of 21,000 RNA-seq data sets called Intropolis, and this could account for another 6% of these junctions. And so we still see about 2.8% of these are novel, and these could still be real, because remember, we're asking for the canonical dinucleotides at the end of the introns to be there. But I wanted you to see sort of a look at the part that I enjoy the most, which is visualizing it and starting to look at what do isoforms look like at the single cell level. And so this is showing a gene that one might think is kind of boring, which is EIF4H, an elongation factor gene. So this is a case where we found something that really kind of blew my mind. You can see at the bottom is the GenCode version 29, and there's a little bit of 5' stutter of different transcription start sites of the first one versus the second. And there's an alternative exon that's down there in the second to last intron. But when we classified these and took a look more closely, if I zoom in just on that 3' end, so the 3' UTR exon, you can see something really strange, and that's that there's actually introns being removed from the 3' UTR of this what I would consider a housekeeping gene. So we can see this in many different tissues. Cell types that were in here, astrocytes, fibroblasts, neurons, et cetera. But what it means is that there's a splicing event that's sometimes taking place in the 3' UTR. And the funny thing is that 3' UTRs, when they do have an intron, it often triggers nonsense decay. And so could this be a gene regulatory mechanism to sort of tone down the amount of this transcript? But that's of course, a question that could be asked now using this type of data. And then looking at cell type specificity, we found that if we only looked at the astrocytes, so the cells that were classified as astrocytes by our parallel short read efforts and clustering, we could see that the tropoelastin gene is only expressed in the astrocyte cells. And so it's known, if you dig in the literature, that astrocytes can make tropoelastin if they're under stress, and the guess is that cells in a dish probably are under some stress. And so you can see from the bottom that GenCode annotates a huge diversity of alternative splicing at this locus. And that includes 5' end differences, alternative cassette exons internally, and also 3' UTR differences, different lengths, different POLYA sites. And so with the single cell information, we could track exactly which isoform is in which cell, and then basically say, this is the isoforms that are being made. And instead of boiling them all together and just saying tropoelastin's on in astrocytes, now this says, no, it's not just on, it's making different isoforms in these cell types. So since we had done chimp and human organoids, we wanted to dig in there and see if we could find anything interesting in that as far as something that would distinguish chimps and humans. And this was a really interesting one that came up, and that's the zinc finger gene ZNF331. And so when we looked in this data, we could see that there's a number of transcripts up above in the chimp cells that are being transcribed from the same spots as the GenCode human transcripts, if you look down below. So these are very common transcripts. But when you look in the human data set, which is the purple, we can see that there's a new 5' exon that's appearing in human that's not seen in any of the chimp data. So these are also annotated in GenCode, but these might have been predicted by a simple liftover that these would also be transcribed from this exon in chimpanzee. But it appears that instead the human is the place that that happens. And interestingly, ZNF331 is one of the genes that by single cell experiments in organoids and in primary cells is a gene that's up in humans, so it's higher expressed in human. And so those studies were done using short reads, and so they would have all just boiled these reads in all together and say ZNF331 is up. But by having this information, it appears that perhaps it's a new promoter that's driving the transcription of ZNF331, and maybe that's why it's up. So finally, I want to sort of go over some of a really recent collaboration we've been doing with Vijay Ramani's lab at UCSF, and that's to evaluate another platform for performing single-cell RNA-seq. And so again, this is one that's been traditionally associated with the short reads, but we thought since this has a similar mechanism of the template switch oligo, we could try and see, could this also be adapted to long reads? And so CELL-SEQ works in a little different way, where there's small micro wells where the cells are loaded into. And then on top of that, beads that encode the oligo DT and the barcode UMI information are loaded in there. And the instrument, the CELL-SEQ Genesis, then makes these reactions happen. And I'll mention that also hot off the presses is that CELL-SEQ is now a affiliate of Biorad. So with Vijay's lab, we took another approach with some mouse neurons. And I'm just giving you a quick preview of these. So again, with the mouse neuron libraries, we see really nice full-length properties. And so these would do even better than our organoid libraries did as far as getting a long full splice match and incomplete splice match in the SCONTI categories when compared with the mouse annotations. And we can actually see even better stretching of the longest transcripts above 5 kb in some of these experiments. And so do these look like real full-length RNAs? So we again took a look at the ends of these transcripts that we're calling, both the TSS that we're calling in the long reads and the POLYA site. And we could see the full splice match, 72% of these full splice matches are within 50 base pairs of a long read start. And also with the novel in catalog and novel not in catalog reads, we're seeing pretty good correlation of these. So reverse transcriptase appears to be doing a good job getting to the 5' end of these transcripts. And these are with mouse primary neurons, so it's quite exciting. And then we look at the other end and say, what's the distance between the POLYA tail in the long read data and where we see those motifs that I spoke of earlier? And you can see that quite nicely, the peak is right around the -15 mark, which is known to be where things are with the terms of cleavage and polyadenylation. And so we're quite encouraged by these types of libraries and what kind of biology that Vijay's lab can do. So today I've told you a little bit about what we're calling single cell iso-seq. So basically taking the cell information, instead of turning that all the way up, try and turn up the information that we get out of these reads. And it looks like about 4 times the isoforms can be found in single cell experiments than in just looking at genes. And so this is with 3 different single cell platforms I talked about today. Any of these platforms that can generate a full-length cDNA and contain the barcoding information can be applied to PacBio. So these are traditionally where they have the information during reverse transcription and a template switch oligo. And so these can be done, and you can also divide the library and do some reading on short reads and some on long reads. And we can determine the barcodes quite accurately directly out of our instrument without cheating off of the Illumina barcodes. And I'll say that it seems like we're just starting to scratch the surface of the cool experiments that you can do with this kind of idea. One, I've talked about the experiments from Hagen Tiltner's lab with mouse neurons and the experiments from Ian McCauley's lab with hematopoiesis. And these have already uncovered really exciting new biology. And I think there's a whole lot of more new biology that awaits when applying this kind of technique. So I just wanted to put up a couple resources that you can go to the PacBio website and get started with your project. We've created a single cell iso-seq library guidelines, and this goes into our normal template prep kit. PacBio we're pretty agnostic to which platform makes the single cell cDNA, and we just want you to get the best data you can. So we say, take that cDNA and go into our library prep. And so there's information on our website for how to do this. If you don't have a PacBio instrument, you can do these experiments and amplify your DNA and then send it off to one of our certified service providers. And these are all over the world. And I've put a picture here of our certified service provider website. You can search for where you are in the world and find somebody that can sequence your sample. And that's you can also reach out to support@pacb.com, which can give you an eye into some of the technical details you might need, or put you in touch with people that can help out, such as me. And finally, I need to give the biggest shout out to my longtime friend and collaborator, Liz Tseng at PacBio, who has been the engine behind the iso-seq informatics. And the tools I talked about today were Sconti, which is an adaptation from Anna Kness's lab that can do these transcript classifications versus annotations. It's been really helpful to go ahead and do some of the QC on these libraries. But it also gives you a lot of other functionalities that I'd encourage you to explore. And then Liz has generated a tool called cDNA Cupcake. She likes to name her tools after foods, which I can get behind when I'm in quarantine here. And that's the package that we use to do the single cell information, like finding the barcodes and UMIs. It can also do things like generating saturation curves, collapsing down redundant transcripts. And so you can go to Liz's website, github.com/magdal, and link into those. And finally, I'll just acknowledge a lot of people that were involved with this work. Besides Liz at PacBio and Brendan Galvin and Ting Han have also participated in the single cell efforts. I've been really grateful to work closely with Evan Eichler's lab over the years, and also Alex Paula and Vijay Ramani's labs. And so last, I'll thank the partners at the actual technology companies, Dolomite Bio, who was so kind to run some experiments with us, both at their headquarters and also right at UCSF. And then recently with CELL-SEQ, it's been really a joy to work with these folks. So please reach out if you have questions. And it's been a pleasure to talk to you today. I'm sorry that 30 kilobases of RNA is keeping us all from talking to each other in person, but it's different times. And I encourage you to still get out there and do your biology.